Let me introduce Christopher Adin, who is Chair and Professor, Department of Small Animal Clinical Sciences, College of Veterinary Medicine, University of Florida, Gainesville. The topic we are dealing with, it's very challenging today, and I'm grateful to Christopher for accepting our invitation. The title of the presentation is Bilirubin as a Therapeutic Drug, Leveraging the Strength of Your Opponent. And we are very excited about this title, and I leave the podium, the virtual podium, to Christopher. Christopher, please start your presentation, and which will be followed by a discussion. Everything is very informal. Thank you.
I'm honored to be here with this group, and a little bit nervous seeing some of the names who are here. Dr. Vitek, I recognize from all of his publications in this area. So please add to and correct me in my presentation. I'll begin by just stating that, I'm a veterinarian, so quite different to many of the people that probably participate in this group. And I thought that it'd be good to introduce some of my background and essentially tell the story of how I've become involved in bilirubin research, because I think that traces well the evolution of the thoughts of bilirubin from toxin to potential therapeutic. And we'll go on and talk about some of the current challenges in enacting the use of bilirubin. So to begin with, I did my surgery training as a veterinary surgeon in UC Davis in the 1990s, and they had an odd situation there where there was an active kidney transplant program in cats. And the reason that that was true is that one of my mentors there had trained in microvascular surgery, actually in a pediatric hospital, and was applying that technique in research, and found that cats, because of their simple major histocompatibility complex structure, were amenable to allograft transplantation with a pretty high success rate and a relatively mild immunosuppressive regime. So it seemed like a good model, and oddly, renal failure is relatively common as cats age, and there were a number of clients that were interested in this therapy. So it also was a good model in that kidneys are paired organs, and there were cats that needed to be adopted from animal shelters, and we could adopt those cats after they donated their kidney. We made that a requirement of the renal transplant procedure. There was some resistance, understandably, to the potential ethical problems with this technique. It was something that I entered as a trainee and started to get a better perspective over time. In particular, European veterinary societies, like the British Small Animal Veterinary Association, were opposed to the use of a donor that we could not consider cats voluntary donors. Even though they ended up being provided with a good home, some animal rights activists felt that it'd be better for them to be euthanized than to be subjected to an unneeded medical procedure. I explain all this because there was great interest on my part in finding mechanisms to obtain cadaveric donor kidneys. But that posed particular problems with organ function, and we needed to really improve our ability to preserve organs from ischemia reperfusion injury. So then I needed to get a job, and I got my first faculty position at the University of Florida. Was subjected to many hurricanes in that time. That's one of them. But I did not have a PhD and needed to develop a research program. My mentor at UC Davis had a recipe that I kind of observed and tried to imitate. I had developed a clinical expertise, but then I needed to develop a research expertise and to find some collaborators that did basic science research so that I could have a leading edge approach to translational research. So I had developed this unique clinical expertise. I could do microsurgery, and in fact, the surgical portion of transplantation in small animals like cats who only weigh 2-3 kilograms is more difficult than doing a transplant in a human being. but I needed to develop a research expertise. And what I decided to invest in was something that, as a clinician, I could understand, and that was the isolated perfused rat kidney model. So this was a model in which a kidney is removed from a donor and then is placed on a machine with circulating perfusate, and the function of the organ can be evaluated during that time. We can measure vascular resistance based on the amount of flow that occurs through the kidney. Urine also is produced, and then you can measure creatinine clearance. You can also add different things to the solutions and perform controlled experiments. So the other important thing in developing a research career as a clinician scientist is that you pick a topic that is important. And I just wanted to mention the importance of ischemia reperfusion injury in organ transplantation, just as one instance. In organ transplantation, it was clear historically that prolonged periods of ischemia led to poor initial graft function. But what was being discovered as I began research in this area decades ago was that injury that occurred to the organ during the ischemic period, when it was deprived of blood supply, led to cellular injury, release of antigens, damage-associated molecular patterns, and then led to a humoral and cellular immune response, and eventual rejection of the kidney was increased. So this was a serious problem. I needed to find those basic science researchers that I could collaborate with, and I was fortunate to find two great collaborators at UF. Anupam Agarwal was a nephrologist who had an active lab, and Byron Croker was a rather unfortunately named pathologist who specialized in renal pathology. And what Anupam was working in was heme oxygenase. He was trying to manipulate the heme oxygenase system to protect kidneys from ischemia reperfusion injury and other types of injury. So this enzyme is known as heat shock protein 32 initially. It has two isoforms, an inducible form, heme oxygenase 1, and a constitutive form, heme oxygenase 2. And interestingly, it's ubiquitous. It's present in everything from blue-green algae all the way up to humans, and therefore was considered to be important. It's induced by heat, stress, the substrates of the enzyme reaction like heme, and also by ischemia reperfusion. And just to see how powerful the effects of that enzyme are, in 2002, this study by Tullius and others showed the effects of 4 hours of ischemia in rat kidneys. On the left is the control kidney histology, and on the right is a rat in which heme oxygenase was induced prior to the ischemic episode. And the difference here is quite stark: a nearly complete preservation of normal renal histology with induction of heme oxygenase, and very severe injury in the control. So heme oxygenase is an enzyme that is responsible for the catabolism of heme, and it produces carbon monoxide and biliverdin. And biliverdin is a structure that is water soluble and relatively non-toxic. And what was confusing is that the physiology of our bodies has developed such an enzyme, biliverdin reductase, that converts this water-soluble and non-toxic molecule into a more toxic molecule that is not water-soluble, bilirubin. And questions are raised as to why that would occur. There must be some reason for forming bilirubin. And these waste products, previously considered waste products like carbon monoxide and bilirubin, began to be investigated. And there's been some interesting research with carbon monoxide that I won't cover today, but that's also a very powerful cellular protectant. Bilirubin was found to be one of the most powerful antioxidants in the serum. In fact, it is the most powerful at low oxygen concentrations based on this landmark study by Stocker and others. It has anti-nitrosative effects. It inhibits leukocyte adhesion and smooth muscle proliferation, making it interesting for myocardial infarction patients. And it's anti-inflammatory and anti-apoptotic. So knowing these things and trying to apply this to my research, I decided to investigate bilirubin as a therapeutic molecule for a variety of reasons. Anupam was working on viral vectors to upregulate heme oxygenase activity. And I wanted something simpler. And as a clinician, bilirubin seemed much simpler to me to enact in the short term. Gene therapy was looked like something that was in the distant future at that time. So we performed a study that was published in the American Journal of Physiology. And this study was very simple. Using Sprague Dawley rats, I removed kidneys and used that isolated perfused kidney circuit to evaluate control kidneys versus those treated with 1 micromolar bilirubin in the perfusate and 10 micromolars of bilirubin in the perfusate. And this group will probably not need the perspective that these are within the physiologic range. 10 micromolars of bilirubin would be towards the high end of the normal range of human circulating total bilirubin concentration. We did 20 minutes of ischemia followed by acellular perfusion. And this is just an illustration of that circuit again. What we found was pretty surprising. There was a stark reduction in vascular resistance in the organs that were treated with bilirubin. So it improved perfusion of the kidneys, especially in the early time phase after treatment. The gold and orange groups here represent 1 and 10 micromolars of bilirubin respectively. And then the other thing we found is that creatinine clearance was significantly improved over time. So organ function was improved in the bilirubin treated groups. Similarly here, the 1 and 10 micromolar groups compared to control. So this was very promising. The other thing that we found with help from Dr. Croker and electron microscopy is that there was preservation of the cells at the renal tubule. So cells in the nephron are involved in active transport and are highly metabolically active. On the left is a cell from the medullary thick ascending limb of the kidney. These are mitochondria that are showing severe degenerative changes. The the membrane of the cell is blebbing. There's a lot of vacuolation and there's pycnosis of the nucleus. So this is a very unhappy cell, as opposed to the similar cell from the bilirubin treated group, which shows preservation of the nucleus, the membrane, and some of the mitochondrial structure, although there is some evidence of injury. So with that promising result, we decided to move on to application of bilirubin in live animals, and decided to use the gun rat model. So this is a model in which rats have mild elevations in bilirubin due to a deficiency or a poor function of UDP-GT, bilirubin conjugating enzyme. And those rats, we administered cisplatin, which is a chemotherapeutic drug that is underutilized. It's highly effective as a chemotherapy agent, but it causes nephrotoxicity and people tend to avoid it. We thought that if we were able to show that bilirubin caused protection, that we could start using this useful drug again. And it was also a good model to try to show the efficacy of bilirubin in protecting against nephrotoxic agents. So Carrie Barabbas, a graduate student in my lab, performed these studies, and we administered a toxic dose of cisplatin and then measured kidney function and bilirubin at days 3 and 5. We used homozygous gun rats, heterozygous gun rats, and then the wild type Wistar. And what we found here, this is the creatinine on the y-axis as a measure of kidney function, and the days after nephrotoxin administration on the x-axis. You can see that the control group showed elevations in creatinine indicating loss of kidney function, where there's complete protection in both the homozygous and heterozygous groups. So this was exciting work. And then at the time, I actually was recruited to move to Ohio State. And this is, if anyone has knowledge of Ohio State, they make people do this horrible OHIO thing every time they take a picture. You can see how pleased I was having just come from the University of Florida, but I felt like a traitor. And I could not find anyone that was interested in collaborating in renal ischemia reperfusion injury research there, to be honest. And this was an interesting move for my research career. What I did find is that there was an incipient islet transplant program there. So they were trying to develop islet transplantation in humans. And I was confused. As a surgeon, I thought, you just treat diabetes with insulin, so what's the big deal? But it is a big deal. Administration of insulin fails to replicate normal physiology, and there is not a tight control of glucose regulation. To the right is some graphs of the blood glucose in patients that have insulin administration. And you can see the wide fluctuations in glucose concentration in their serum or their blood. And then on the bottom, after islet transplant in these same people, a much more tight regulation of glucose. This leads to elimination of the progressive aspects of diabetes, which continue to occur with insulin therapy. The other thing that's important is that this is a big disease in veterinary medicine as well. I won't bore you too much with that to this audience, but about half of the dogs that are diagnosed with diabetes are euthanized at the time of diagnosis because the veterinarian explains the need for lifelong daily administration of insulin, monitoring of blood glucose, and owners feel that the quality of life and the long-term need for therapy are not compatible with their goals for their pet. So you can do organ transplantation for diabetes, and pancreas transplantation is possible, but it's complicated. And there's a lot of plumbing involved with the pancreas, which makes it more complicated than kidney transplantation. And most importantly, the anatomy of the pancreas is such that 99% of the pancreas is involved in non-insulin producing function. It's a digestive organ. And all of this tissue that's being transplanted is unnecessary in most of the patients that receive pancreas transplants. They're getting it to treat diabetes, and they're getting a whole bunch of digestive tissue that's unnecessary. So islets are these small clusters of cells, about 1,000 cells in a large islet, and they contain cells that produce a variety of endocrine substances, one of which is insulin. The beta cell is the particular one that's the most desired in transplantation. And it has a radial blood supply. So interestingly, while we're doing cellular transplantation of islets, it's somewhat similar to kidney transplantation in that it does involve the deprivation of blood supply to the transplanted tissue. And unfortunately, unlike kidney transplantation, we don't restore that blood supply when we transplant cells. Those cells have to develop their own blood supply in the new location. So they're actually subjected to a more prolonged period of hypoxia than with an organ transplant. And that is the main problem in cellular transplantation for diabetes. So again, want to make sure that we're dealing with an important problem. And 1 in 10 human healthcare dollars are spent on diabetes mellitus. So this is a huge need. If we could cure diabetes with cellular therapy, it would potentially be even more important than kidney transplantation in changing the face of medicine. The other things that are ripe for adoption here is that it's the deficiency of a single small molecule. So it's relatively simple. We only need the transplanted cells to do one thing. They need to sense a single small molecule, glucose. And the reason I say small is that the transplanted cells can be placed in a nanoporous device of some sort that prevents access of immune cells, but allows this small molecule to travel through the pores. So how do you get islets isolated? Just briefly, we typically digest the pancreas using enzymatic methods to free the islets, which are stained here with dithazone, from the pancreatic digestive tissue, and then we discard that. And then islets are transplanted through injection into the portal vein, typically in people, and then it will seed into the liver. And the thought is that those cells are in a relatively normal position and in terms of exposure to the blood that's emanating from the intestines, much like they would be in the pancreas. So the biggest problem with islet transplantation, as I intimated earlier, is hypoxic injury to the cells. Because they're non-vascularized, they depend upon diffusion, and what tends to happen is that the center of these large clusters of cells becomes hypoxic, and those cells will die. And this is a simple live dead stain technique showing the death of cells at the center of the islet. So 50 to 70% of the cells are actually lost in the first 24 hours, and this leads to devastating problems with islet transplantation. It essentially has made it not widely adopted, despite all of the advantages I just talked about, because a pancreas, which is donated, is extraordinarily valuable. And if you can do a pancreas transplant through organ transplant techniques, you have a reasonably high success rate. People would much rather avoid the complex surgery and get a cellular transplant, but if you lose 50 to 70% of the cells in the process, most people end up needing multiple islet transplants or multiple pancreases before they can reach a state of improved function. And that just is a no-go. So it's been hugely problematic to this developing area. So this is an interesting model, I thought, to study, in that the islets, you can maintain them in culture, and you can subject them easily to different conditions without administering, without having the entire body to worry about. So it's an isolated system. And it lacks the complicated ultrastructure of the kidney and the potential surgical problems due to me that might affect the outcome if we're doing experimental studies. So the first study that I saw when I began looking into this involved the use of bilirubin to induce tolerance to islet allografts. So interestingly, this group showed that the administration of bilirubin not only improved function of islet grafts, but it induced tolerance, immune tolerance to the organ. And this was really interesting to me. It meant that it probably was reducing the production of antigens by the injured cells and therefore preventing recognition of the transplanted tissue. And this led me to read more about the danger hypothesis. This is something described by Polly Matzinger, that the immune response to the allograft is determined by cytokine expression and damage-associated molecular patterns from the transplanted tissue rather than self or non-self recognition. And it really doubles down on the importance of protecting organs and cells in transplant. So my hypothesis was that exogenous bilirubin supplementation would induce allograft tolerance by decreasing cell death and alarmin signaling in transplanted islets. And I was fortunate to get some funding for this through the CTSI, Clinical Translational Sciences Institute at Ohio State, and did some research training under a KL2 award there. So this is just an illustration of the mechanism that injury to the islets causes an immune response by triggering the damage-associated molecular patterns to be released. One of those would be HMGB-1. It also induces toll-like receptor signaling. Reactive oxygen species can contribute, and it induces the inflammasome, and that leads to production of interleukin-1 beta. So the first study that I performed in this area was related to administering physiologic doses of bilirubin to islet allografts. Actually, we did this in vitro at first, and I worked with some colleagues who were involved in pathology and basic sciences to help me with some of the molecular level techniques that we used. The model, again, was relatively simple. We had AJ mice that were controls. AJ mice that were administered 2 micromolar per kilogram bilirubin intraperitoneally, and then control islets versus islets that were administered 20 micromolars of bilirubin into their culture solution. So we had both administration to the animal and administration to the islet media. We did standard islet isolation and then exposed those islets to hypoxia, and then evaluated viability of the islets. We did an ELISA on the media to look for evidence of the damage-associated molecular patterns that may be released, and then did PCR. So hypoxic stress here is the effects of hypoxic stress on cell death are illustrated here, with cell death on the y-axis being indicated in percent. You can see that the control islets had about 40% cell death after hypoxic injury. Compare that to the mice treated with bilirubin intraperitoneally, a reduction in cell death, and actually most powerfully, administration of bilirubin to the media for the cells caused the greatest reduction in cell death. And this is great because it's difficult to administer bilirubin to a patient prior to islet donation, but much easier to administer it to the cells after their ex vivo. So a little bit on the toll-like receptor pathways. TLR2 and 4 are stimulated by damage-associated molecular pattern molecules like HMGB-1 and HSP70. And then that primes the NLRP3 inflammasome with interleukin-1 beta. So we evaluated some of these substances, but I wanted to just mention that TLR2 and 4 are involved in injury sensation and they mediate early graft function after transplant. So we measured HMGB-1 and HSP70 as a result of our knowledge of those as common DAMPs. And we found that those results paralleled the cell death results that I showed earlier. This is the fold change in HMGB-1 compared to control control prior to hypoxia, control after hypoxia. You see there's an increase in release of this marker, and that is prevented by bilirubin being added to the media. Similarly, with HSP70, adding bilirubin to the media prevents release of this damage-associated molecular pattern. So we wanted to just document that reactive oxygen species may be involved in this mechanism, and we measured reactive oxygen species in the islet media and decided to do that using 8-iso-PGF2 alpha ELISA. So this is a relatively simple technique that we use in our lab. We found, as expected, that in the control group, the evidence of reactive oxygen species was increased by about 100%, and that was prevented by addition of bilirubin. So it did appear that the positive effects were concurrent with a reduction in reactive oxygen species, and that bilirubin may be acting through its antioxidant mechanism. So I think I've talked about this enough. We did show that interleukin-1 beta expression was reduced, and that helps to indicate that it may also be involved in reduction of the activity of the inflammasome. I keep moving along. So the current understanding here is that bilirubin inhibits the release of damage-associated molecular patterns, that it's a powerful antioxidant, and that it seems to be working within this islet transplant model, that it suppresses the inflammasome, and that it is an immunomodulator as well, based on these previous studies inducing tolerance. So now that we've got some background on the experimental work that I've done, which is quite humble compared to some of the work that this audience has done, I wanted to talk a little bit about the evidence of efficacy of bilirubin in the human population. So this is one of the most exciting things about bilirubin research, is that we have existing models in the human population. Unlike most drugs or small molecules that we're trying to investigate, we already know that it works. We don't have to have clinical trials necessarily. And that's through the existence of Gilbert syndrome, which is present in 5 to 10% of the population. It's a mutation in the promoter region of UGT1A and leads to mild elevations in serum bilirubin. What we know is that there's a 67% reduction in the risk for coronary artery disease or myocardial infarction in people with Gilbert syndrome. There's also a reduced risk of obesity and metabolic syndrome, or conversely, if you have low bilirubin, you have an increased risk of those conditions. And that there's improved outcomes in organ transplant in people that have elevations in bilirubin. So really excitingly, we already have evidence that elevation in serum bilirubin, if it could be achieved, would be beneficial in the population. So this opens up ideas for therapeutic applications of bilirubin in many ways. It could be applied in diabetes and metabolic syndrome, in stroke, in organ and cellular transplant, in the therapy for diabetic ulcers, and in myocardial infarction. And there's been some wonderful work in all of those areas, which I don't have time to summarize today, but again, I think some of the people here have contributed to that. What I wanted to talk about for the rest of the program was the problems in applying bilirubin. We know that it works. I feel that it works based on the evidence that is available, and I don't have time to present all of that today. But the problem is that the bilirubin that is widely used in these studies is animal sourced. It's typically porcine derived, and there's a variety of companies that sell bilirubin. There's been some literature decades ago about the differences between those products as well from each company, the potential endotoxin that may be contained in it, levels of purity. It's typically composed of mixed enantiomers. And as a result of all these characteristics, it's not the kind of thing that the FDA would like to approve. To top it off, physicians are fearful when you talk about administering bilirubin to their patients, because it's something that they've had as a foe for most of their careers. Lastly, it's poorly soluble in aqueous solutions, making it difficult to administer as a drug. So the rest of the time, I'll talk about some interventions that we've been trying to work on to address some of these shortcomings. So the various approaches to bilirubin delivery, one would be induction of heme oxygenase. As we talked about before, that was something that Anupa, my colleague at UF, was trying to work on. You can administer heme or myoglobin as a substrate of heme oxygenase, but these substances are toxic in themselves, and you also need to administer it prior to the episode. So this really limits the number of diseases that you can use it in. You have to have planned ischemia rather than unplanned. You also, we basically haven't found a non-harmful chemical that can be used as an inducer, although this would be an exciting area if it could be identified. There have been some chemicals or drugs that have been known to inhibit UGT-A1A conjugation of bilirubin. So if you can inhibit the conjugation of bilirubin, you can increase serum bilirubin levels. One that's known is atazanavir, which is an antiviral drug. And this is something I was interested in, I think probably 10 or 15 years ago. I noted in the literature that it was a side effect of that drug. And people hadn't really focused on using it as a therapeutic. And I think only recently have people come around to think again about applying it for its side effect, essentially, to elevate serum bilirubin levels. Curcumin is another molecule that can cause elevation in serum bilirubin levels through inhibition of UGT-A1A. Another method of addressing some of these issues in drug delivery is nanoparticle encapsulation. So with this technique, you can target certain cell types because certain cell types have a higher rate of phagocytosis, and by adjusting the size of the particle, you can increase uptake by certain types of cells as well. This can also be a way to tune drug release because based on the molecules that you use or the polymers that you use for the coating, you can adjust how quickly the drug will diffuse through. So you can have prolonged administration of the drug after giving nanoparticle encapsulated bilirubin. The other thing is that you can increase water solubility. So I pursued this at Ohio State with an engineering colleague named Xiaoming He, and he helped us to do nanoparticle encapsulation of bilirubin. What we did in that study was we used pluronic F127. So this is a polymer that has amphiphilic characteristics where it will have a hydrophobic center and a hydrophilic exterior that these micelles will improve solubility of non-soluble compounds like bilirubin or molecules like bilirubin. So we did a study with insulinoma cell line, which we found easier to manipulate. We cultured them in sterile 35 millimeter Petri dishes, and we wanted to just test for toxicity of these nanoparticle drugs and also the ability to protect from hypoxia. We exposed these INSR3 cells to hypoxia after providing them with either free bilirubin, empty nanoparticles, or bilirubin loaded nanoparticles at various concentrations. And then we also used lysotracker red. This was added to the media to allow us to track the phagocytosis, and we figured that the bilirubin would be located and the nanoparticles would be located in the lysosomes. So this is the cellular uptake experiment where we wanted to see how nanoparticle encapsulation altered the uptake of bilirubin. We used fluorescence microscopy. This is empty nanoparticles, free bilirubin in the solution, and nanoparticle bilirubin. The lysotracker red shows the lysosomes. Bilirubin is green fluorescent. And then when you superimpose them, you can see that the uptake of bilirubin was actually higher in the bilirubin nanoparticle group in these insulinoma cells than it was in the free bilirubin group, which is interesting. So it does appear that nanoparticle encapsulation subjectively increased uptake. The next thing that we did is we measured its effect on cell viability. And here we have the cells that were not treated. The viability was 100%. When we treated with nanobilirubin at 5, 10, and 20 micromolar, we found that at higher concentrations, the cell viability went down, interestingly. So there does appear to be some toxic effect. And then you can see the same graph for free bilirubin and empty nanoparticles, which also seem to have pathologic effects on these cells. The one concentration that didn't seem to cause harm was the 5 micromolar concentration of nanoparticle bilirubin. Now, this is the cells after they were exposed to hypoxia and pre-treatment with either 5 micromolar, 10 micromolar, or 20 micromolar bilirubin. And here there's nanobilirubin, free bilirubin, and empty nanoparticles. And the illustration to the left shows a graph of the cell death. And what you see here is that cell death was lowest in the nanobilirubin treated group at 5 micromolar. So that appears to be the most effective therapy compared to free bilirubin and empty nanoparticles. And this is the illustration of the cells. You can see at 5 micromolar, there's the lowest amount of cell death, which is illustrated by the pink staining cells. So under hypoxic conditions, nanoparticle bilirubin appears to be the most effective. Lastly, we measured islet function, and we used a technique called stimulation index, which compares the amount of insulin that's produced by an increase in glucose concentration, so sort of the cellular response to an increased concentration of glucose. The control islet cells had about 3.5 stimulation index, and you can see that nanobilirubin preserved that, whereas in the empty nanoparticle and free bilirubin group, we did not reach that level of islet function. So this approach only deals with some of the problems of bilirubin therapy. It only really addresses the delivery of the drug. We were still using bilirubin that was sourced from pigs, and it was not ready for use in humans. So we had a long way to go. The last thing I'd like to talk about is our work in trying to develop a synthetic form of bilirubin that would avoid those problems with being animal sourced. Ideally, we really wanted to develop a synthetic bilirubin that was water soluble and was not derived from animal sources. And we succeeded to some degree, but not in all of our goals. In this work, I was at North Carolina State at the time, and I worked with a synthetic chemist named Josh Pierce. And what they developed are some simpler versions of the bilirubin molecule by pyrols. And this is an illustration of the two reactions. We called them compound 1 and compound 2. The second one that he developed was a little bit more complicated molecule to try to simulate the structure of bilirubin more closely. So these are not tetrapyrols like bilirubin. They're a simpler version that have some similar chemical characteristics, but are easier to synthesize. We worked with a laboratory that did antioxidant profiles of various natural substances. And we found using the FRAP and ABTS methods that bilirubin was comparable to quercetin in these studies. Bilirubin is indicated by the triangles here. Compound 1 is relatively close to bilirubin in its antioxidant capacity. And what you notice here is that the concentrations are quite high on the x-axis. If you look down in the range that we were using in our earlier studies, bilirubin is actually, the compound 1 and bilirubin are actually closer to quercetin at that level. And in fact, compound 1 is superior. Similar results with ABTS. Compound 1 appeared to be an effective antioxidant, whereas compound 2 did not appear to be as effective in either of these comparisons. So we decided to move forward with compound 1 in further studies. First, we wanted to look for evidence of toxicity. So we administered increasing concentrations of compound 1, 10, 20, 40, and 80 micromolar percent cell death here, and this is free bilirubin in the solution. We found that at 10 micromolar, there was no effect on cell death, but at higher concentrations, both compound 1 and bilirubin did induce death in these islets. After hypoxic injury, we also wanted to show the protective effects of this compound. What we found is that compound 1 at 10 micromolar decreased cell death compared to control islets significantly. At 20 micromolar, it did not. And then it was actually more potent than free bilirubin in the solution when we used in this particular study. So I think I've already showed that slide, but I think this is the last exciting step that we've made. I don't think we're quite there yet, but I did want to try to stimulate discussion in the time that we had remaining from the brilliant minds that are here. Bilirubin is a potential therapeutic and some of the most impactful conditions in human medicine. There's evidence of efficacy already, which makes it exciting. Little work is really focused on harnessing the therapeutic effects. I think everyone has focused on experimental studies, and few have tried to transfer this into human therapies. I think I am not knowledgeable in the business world, but I presume that some of that is fear of being able to actually license a drug that could profit a company. The opportunities abound. So I'd love to stop there and have some time to discuss things.
Okay, Christina, you want to take over? Hello? Christina, can you hear me?
Yes.
You want to take over?
Yes, I'm very sorry, but I have some problems with my connection. So hi, Dr. Adin, and thank you very much for your presentation. Very interesting, very clear, very incisive. And I thought the most interesting aspect is to try to put together the clinical expertise and the basic research expertise. And also to try to face this problem of that bilirubin. We know that bilirubin is protective because we know that Gilbert subjects are very lucky, but we don't know how to extend this protective aspect to the general population. That is something that probably we can do, but we don't know how to do, and probably this is the way to follow, to work in. So thank you, and I would like to ask all the participants if there are some comments, some questions, please.
So may I have a question, Christina?
Yes, sure.
Oh, good. So Christopher, first of all, thank you very much for your talk. It was very educative, and it showed us, as Christina said, that it's a great mixture of basic science and clinical experience. So I have actually two questions to you. One is related to your islet, pancreatic islet transplantations. I'd like to ask you whether you try to go into deeper details in terms of metabolic pathways which might be affected by bilirubin. I'm talking about, for example, agonistic effects on insulin receptor or AMP kinase or PPAR alpha or PPAR gamma or all these pathways. So this is one question. And the second one is related to the last part of your talk, because you talked about this dipyrolic compound, which is very interesting. Our own project related to the biosynthetic pathways, which should result into the preparation of either monopyrolic or dipyrolic human body. I'm talking about those propendiopen's or even tripyrolic compounds. So why, in other words, do you think that your dipyrolic compounds could be really used as a medicine? Because if they are not, let's say, biologically occurring in a human body, it might be a problem, in my opinion. Whereas if you would use those which are physiologically present under certain conditions in a human body, it might be much more easier. So could you comment on that?
Yeah, thank you, Dr. Vitek. I think both of your comments are show your deep level of knowledge in this area. And the first one, we did not do those molecular level effects. We did not investigate the molecular level effects in the islets. I think, and your work and others in doing that at the systemic level in animals or humans is really interesting. I think the islet model might be something that you or others could think of using. I don't have that expertise, to be frank, but the islet model is simple because the cells are isolated. It may not reflect the whole picture, but at least it is a piece of it. And I think it's probably ripe for mechanistic studies like that. So I'd be interested in talking more about that.
Okay. So it might be a space for some collaboration.
I think so. Yeah. And I'm not particularly unique in having the ability to isolate pancreatic islets. So I'm sure you can find someone close to home, but I'd be happy to help from afar. The second question, I think your biopharmaceutical approach is a really smart one. And I agree that this molecule that we evaluated is far from ready to apply. And I think part of the struggle I have as an academician is that, I just don't have the, and maybe this is a cop-out, but I don't have the system set up to take the discoveries that I've made forward. I have done the part that I can, but the next steps in terms of getting a product evaluated through safety and efficacy testing would probably need to be taken up by a company that is interested. And that's not, I have not approached this from a commercializing approach, and that's probably a negative. Anyway, I think that you're correct, and that there'd be many steps needed in order to make sure that this was a safe compound.
Okay, thank you. Yeah, it would be very interesting to see whether your compound is also a compound which can react with any of the receptors on the cells or in the nucleus, because
yes,
it's a big topic now.
Yeah, that'd be something I'd be happy to discuss. We can certainly make more of the compound with Dr. Pierce's lab, and share that with anyone that's interested here. We haven't licensed it at this point, and I do want it to go forward if there's something useful to come of it.
Good, thanks.
Thank you.
Please, Sylvia.
Okay,
I have a question.
Thank you, Christina. Thank you very much, Christopher. I really enjoy your presentation, mostly because I'm also working on the protective side of bilirubin. And we proved that bilirubin, simply given to brain organotypic cultures, can protect from dopaminergic neuron loss in our model of Parkinson's disease. So we also have the same question, how to deliver bilirubin to a human. And we also moved to nanoparticles loaded with bilirubin. But my question is about your compounds. Do you have some idea, or you can think that your compounds can enter the blood-brain barrier?
That is a good question. Unfortunately, these compounds are still not soluble in aqueous solutions. That I forgot to mention that in the talk probably, but that was one of the things we failed to achieve in working with the synthetic chemist. So we had to take all the same measures, dissolving in a basic solution and then titrating. So I think that it would probably be similar to bilirubin in that regard. And I would have to defer to my chemistry colleague in terms of the characteristics of the molecule. So but we didn't test that either.
Yeah, but your opinion is probably that for delivering your compound to the brain, you need again a vehicle, a nanoparticle, something like that to cross the blood-brain barriers.
It's likely that you'd have to take similar approaches to free bilirubin. The only thing that was accomplished in that study was that it's synthetic rather than animal derived.
Yeah. Okay. Thank you very much.
I think Professor Tiribelli have a comment.
Oh, John, hi. You want to comment, John?
Yeah, no, I had a quick question for Chris. Thanks so much, Chris. I really enjoyed your perspective and insights. So my question, maybe what Claudio is going to follow up on, is related to the concentration of bilirubin exposures that you had to islet cells and some of your other in vitro studies were what I would perceive is fairly high, even the 2 micromolar. So, but they apparently weren't toxic. So I was interested in what was the composition of the media, and something else must be binding up some of the bilirubin. And did you measure the free bilirubin levels, unbound levels in your in the media and during those exposures?
Great questions. Yeah, so we use RPMI 1640 for the islet media, and there is fetal bovine serum added as well. And I think it's probably binding to albumin in the media. So somewhat similar to a physiologic system, I guess, without all of the complexity. I think in an article that I worked on with Dr. Bellarosa recently, I tried to go through the history of the dosing of bilirubin, and it's very interesting. In my own lab, we've had differences between the amount that needs to be administered to an animal versus to the media. And I think part of that might be the conditions like the presence of albumin in some of those experiments and the lack of it in others. But we've found that if you give the same dose to an animal that you give to in a cellular culture, you don't have the same effects. And we found that we needed higher doses in vivo compared to in vitro, to sum it up.
Thank you.
Well, let me take on what John just asked. I mean, I think there is a way you can roughly calculate the BF, knowing the amount of albumin present in the medium, and whether albumin is human albumin or bovine serum albumin, because the binding affinity are totally different. There is a paper I advise you to go through. It's published in Pediatric Research, and the first author is Rocca, and the last one is, the second one is Caligaris, and they provide some nice charts by which you can really have an idea of the BF, which actually is the key point in making experiments. But let me ask you, I was really excited by your presentation, congratulations, because actually, as stressed by Sylvia, bilirubin now is something much more than it's simply a yellow waste product. I mean, we know that bilirubin is a sort of, acts like a hormone. Together with Libor, we provided a recent review in Journal of Pathology suggesting that bilirubin is an hormone. But the question is then, bilirubin is very difficult substance to work with. It's sticky, it's water insoluble, it's light sensitive. So we need to find out substitutes. And your compounds looks very much promising. Unfortunately, your compound is still insoluble in water, which actually makes the compound similar in terms of difficulty to work with bilirubin. But if we can solve the issue of water solubility, I strongly advise you to check your compound on some more, let's say, close to reality molecular models. I think the model Sylvia is working with, which is neuron slices, may provide some information much more quickly than a cellular or a in vivo model. And then you can also follow in this model all the effects provided by the compound, stress and inflammation. Because as far as antioxidant, it's true that the bilirubin is a potent antioxidant, but it's not the only aspect. I think it's much more important the anti-inflammatory effect than antioxidant. In addition to what bilirubin per se is doing inside the cell, all the homeostasis of calcium and you name it. I think, your way, it's very tricky and very important, but this provide the possibility to remove the nanoparticles, because nanoparticles are a nice mean to deliver bilirubin, but are rather difficult to work with. As Sylvia knows, being involved in this kind of stuff since more than 1 year. And I think that nanoparticles have been demonstrated to be effective. Libor and I wrote a sort of editorial in New England Journal of Medicine on the different models. But if we can get something which is much easier to work with, that's what we're being done.
Yes, thank
you. That's my comments. Congratulations, that's nice. If you need any help in order to prove or disprove your thesis, I'm pretty sure that I'm also able to talk on behalf of liver, since we are at very close labs together, let us know. I mean, it would be fun to work together.
Thank you. That would be wonderful. To be honest, my laboratory has ground to a near halt, given I've taken on a department chair role now, and I would love to to hear ways that we could keep this research going forward. If not this compound, maybe the process that the synthetic chemist described will be helpful in stimulating others to think of similar compounds that can be synthesized.
Okay. Let me finish one additional point. As I mentioned before starting the conference, I was in Gainesville ages ago, but I noticed that you work in Davis so probably you met there Dr. Richard Wember, which is actually one of the fathers of the free bilirubin story. And I think that Richard is a good friend of us, and he spent 2 years with us. And the yellow world is rather small.
That's funny. Yeah, I seem to have been traversing all of the hotspots for bilirubin, so I'm very fortunate, obviously, and I really feel honored to have been part of this group today. Hope to continue to tune in. I apologize that I haven't been able to this point, but I would love to listen to future discussions.
You will also be welcome. Christina.
Thank you very much. Are there other questions? Or no? Thank you, Christopher. We hope that yes, this conversation will continue in the future and with a higher collaboration and discussion.
Let me finish saying that now we are moving from China, where someone is going to sleep, to Florida, where someone is going to have a breakfast. So it's really a large word related to bilirubin. How, John? Bye-bye.
Bye-bye. Thank you.
Ciao, John. Bye-bye.
Thank you.
Thank you. Bye-bye.
Ciao.